Good afternoon, everyone. I am student Dr. Vi Nguyen. Over the summer, I've had the pleasure of working as an NIH fellow in Dr. Staudinger and Dr. Kramer's lab. Uh, just working or moving forward with what Whitney has done, my study focuses on the effects of cordyceps extracts on lipid and drug metabolizing enzyme expression on human colon cancer cells. So what are cordyceps? Cordyceps are parasitic fungi that produce cordycepin and other bioactive compounds. These are some of the bioactivities of cordyceps, including existing literature on its hypolipidemic, hypoglycemic, and anti-inflammatory effects. They're not your typical mushrooms. They grow on larvae of insects. Here are different species of cordyceps. This is a zombie ant that has been infected, lab-grown cordyceps. And if cordyceps sound familiar to you, you might have heard it from the HBO series, The Last of Us, in which, you guessed it, cordyceps was responsible for the zombie outbreak. The cordyceps shown here is the exact extract we use for our study. It's very accessible and can be found at your local health food store. It's advertised as having health benefits as well as providing energy. Moving on to the methods, I treated LS1 colon cancer cells with cordyceps for 48 hours. The different treatment groups includes vehicle and 2 doses, which is 25 and 100 micrograms per milliliter. Our TQ-PCR was performed as well as a reporter gene assay. The goal of my project was to identify which genes get zombified or upregulated and silenced or silenced. The different genes I focused on are categorized into 3 panels, which include cell cycle, drug metabolism, and metabolic regulators. Here is a brief overview of real-time qPCR. After treating the cells with cordyceps, I extracted the RNA, transcribed it to cDNA, and after fighting my lab mates for the qPCR machine, I was able to get some hot data. The hot data are on 30 different genes. These genes are divided into 3 separate panels. In the first panel, panel A, studies cell cycle. Proto-oncogenes are normal genes that, when mutated, can become cancerous and promote cell growth. On this graph, as you can see, these are my treatment groups, and on the x-axis are the different genes I investigated. So there is a downregulation of these proto-oncogenes, as well as tumor suppressors, which suppress growth. There is a downregulation except for TP21. So what does this tell us? It implies that cells are receiving fewer go signals, making cells more prone to arrest or death. However, there is a limitation. Throughout the cell cycle, there are different stages, and depending on which stage the cell is in, the protein levels can vary from one stage to another. For the next panel, panel B, these genes study drug metabolism. These are further split into 3 subcategories. For the first one, there is an upregulation of genes responsible for membrane transport. So we're talking about transport of lipids, bile acid, bilirubin, and drugs. For the second and third one, there is an overall upregulation of drug metabolizing enzymes as well as conjugating enzymes. So what does this tell us? Well, cordyceps upregulates or zombify key drug efflux and bile acid pumps, as well as drug metabolizing enzymes. These findings signal a pharmacokinetic warning. If cordyceps induce these pathways, it can accelerate the clearance of drugs taken at the same time, potentially reducing their effectiveness. This is important because these are very accessible. So if a patient goes into a store and they decide they want to start these supplements, they might not be aware of how it could affect the other drugs that there are also prescribed. For panel C, these study metabolic regulators. As you can see, There is an upregulation of these genes involved in carbohydrate metabolism, but there is a downregulation in PepsiK and upregulation of lipid handling genes as well as genes involved in ketogenic shift. So the results imply that there is a metabolic rewiring occurring. Cordyceps downregulate PepsiK, which is involved, a rate-limiting step involved in gluconeogenesis. Their cordyceps upregulates these genes involved in mitochondrial activity as well as glycolysis. This supports existing literature that cordyceps help regulate blood glucose levels by reducing glucose output as well as increasing glucose breakdown. In addition, cordyceps upregulates this gene that is involved in lipid metabolism. Also supports existing literature that cordyceps may play a role in lipid management, most likely cholesterol lowering. This cordyceps upregulate PDK4, which is involved in fatty acid oxidation. So there is a shift in cellular metabolism away from glucose utilization towards fatty acid oxidation as an energy source. However, there is no current literature supporting cordyceps' role in ketogenesis. Our results shows that there's an upregulation of HMG-CSH2, which is a rate-limiting enzyme in the breakdown of fatty acids into ketone bodies as an alternative energy source. In summary, cordyceps is super bioactive. Our data opens up a new angle that we can dive into, especially with ketogenesis. In our last study, we performed a reporter gene assay on these nuclear receptors. So these are the nuclear receptors that we investigated with and without PGC1 alpha, which is a co-activator. So we transfected the cells with plasmids, and one of the plasmids had our gene of interest. We treated the cells with cordyceps, and we were able to measure the luminescence. So these are our results. As you can see, there is an upregulation of FXR, which is involved in bile acid metabolism, an upregulation in GR and LXR, which is cortisol-mediated stress responder, as well as cholesterol efflux activator, as well. And there is an upregulation in PPAR alpha and PPAR gamma involved in fatty acid oxidation and adipogenesis and insulin modulator. And this graph just kind of condenses everything with the co-activator. There was only a significance with GR with PGC1 alpha. Overall, cordyceps, an over-the-counter supplement, is super bioactive and have many health benefits, but it can reduce prescription drugs efficacy due to its herb-to-drug interaction. I want to take this moment to acknowledge those who made this possible. I've had an amazing experience with amazing people, and I'm truly grateful. Thank you. Any questions? Yes.
Can you hear me? What does the taking cordyceps have to do with colon cancer? Does it help treat colon cancer?
So we use colon cancer cells because it's a good model to study gene expression levels in. These just studies the effects of, so for qPCR, oh, sorry, let me go back to the data. So these are all, these cells are treated with cordyceps, and we're trying to see the effects of cordyceps it has on colon cancer cells. And based off of that, we could maybe move forward to seeing how these proteins interact with each other in order to maybe develop future drugs that, yes, Jeff,
So
Dr. Staudinger.
let me add to that really quick, and that is, if you think about oral administration of mushrooms, what's the first stop? Your stomach and then the gut, so colon. It's also an excellent model to study the effects of that. So if you think about a whole person, one of the first tissues that's going to be exposed to this is colon. So that's the other rationale for using this cell line.
They also express PXR endogenously.
So no need for forced overexpression.